So we're going to turn our attention now to another key figure in the story of the germ theory, and this is Robert. It's spelt Koch, it's pronounced Koch, like a male chicken. And he is a contemporary of Louis Pasteur's. Now we've seen that in 1861, Louis Pasteur develops something called the germ theory. And by 1878, Pasteur is confident enough to publish, along with associates, the germ theory of infection. So 1878. Or disease, sometimes it's known as the germ theory of disease. And this makes a clear link between germs and infection and disease. But Pasteur isn't all that effective when it comes to identifying the different germs that cause different diseases. And this is quite important. One of the problems that people have when it comes to accepting the germ theory is that when you look in the human body, and particularly in places like the gut, there's quite a lot of germs and bacteria, and people would say, well, if these are present in a healthy person, how can they be causing disease? And this is where Koch comes in. Now, Koch is German, Pasteur is French. There's actually quite a lot of rivalry between them, partly because France and Germany go to war between 1870 and 1871 in the Franco-Prussian War. Germany wins, but both are kind of motivated to prove that their country is ahead. Now, there are two methods that Koch develops that really help in the process of identifying germs. One is that he develops a method of growing germs in agar in a petri dish. So that makes them easier to sort of isolate and identify. So methods of growing germs in agar in a petri dish. So that's important. The other technique that he develops is the staining of germs with industrial dyes. So I'll do this with a paintbrush, which is not literally how they did it. But Koch develops a method for staining different germs, and this means that germs are easier to differentiate between under a microscope, and it's easier to identify the germs that are specifically causing specific diseases. So for example, in 1876 even, Koch identifies the anthrax bacteria. And then Pasteur applies the principles of vaccination and develops a vaccination for it. Pasteur has also developed a vaccination by this point against rabies. In 1885, he saves a 13-year-old boy who's been bitten by a rabid dog by giving him 1885 is round about here, by giving him 13 injections over a 2-week period. Koch then goes on to identify the TB bacterium, the bacteria that causes tuberculosis. So 1882, and this is considered really difficult because it's such a small germ. And by this time, other scientists are getting really interested, and there's kind of a rush to identify the different germs that are causing different diseases and to come up with solutions to solve those diseases based on this knowledge. So, for example, in 1883, they identify, which is just a little bit further on from that one, they identify the diphtheria bacteria. Diphtheria was a horrible disease that often attacked children, would cause a high fever and a cough, but it made a kind of skin grow at the back of their throat so that they couldn't breathe. And they discovered that diphtheria was producing a poison that settled in the back of the throat. So they were able to identify diphtheria. And because of that, by 1913, they had a vaccine for that. And so it goes on. By the 1900s, they have identified the germs that cause tetanus and polio and dysentery and meningitis and plague. So a lot of horrible diseases that have seemed unstoppable in the past are being identified, and as a result vaccinations are being developed. So we've mentioned the diphtheria vaccination in 1913. There's a tetanus vaccination in 1927. There's a TB vaccination in 1906. So this is a really exciting time in medicine because they're able to identify what is causing disease, and they're also able to come up with ways to prevent it, if not to cure it. Now in 1910, there's another breakthrough that is caused by one of Koch's students, whose name is Paul Ehrlich. And he is able to identify a chemical that specifically kills the bacteria that causes syphilis. And what this means is that they can then produce what's known as a magic bullet drug. A magic bullet drug is a medicine that you can take that will deal with the cause of the illness without having too many side effects. And this one for syphilis is called Salvarsan 606 because it was the 606th chemical that he tried to do this. So in addition to developing vaccines, people are finding that they're able to develop treatments now for disease as well. So this is a very dynamic and a very exciting and a very helpful time. The knowledge of germs has a lot of results. So for example, knowing about germs leads to great developments in drugs and chemicals to kill them. It leads to great developments in aseptic surgery because once people understand that it is germs that are causing infections in surgical patients, they're able to target it. And it also has a big impact on public health because once governments begin to understand that germs are causing disease, then it's easier for them to understand how to target them. The British government is not particularly receptive to germ theory. So for example, in 1884, Pasteur actually went to Calcutta in India to research cholera, and the British government just ignored this and said, no, it's miasma, despite the problems that cholera was causing in Britain at the time. So, although some people are slow to accept it, ultimately the understanding of germs has a huge effect. The other impact of this is that people stop studying symptoms and they start to study diseases instead. So after years and years and years of not really understanding why people get sick and what happens to their bodies when they get ill, all of a sudden, scientists and doctors have a much clearer picture of what is going on.